Hi, welcome back. This is the continuation of Chapter 11, Part 2. This is a little bit longer. I will at some point tell you we're just reviewing from Bio 1, so if you're feeling very brave, you can skip that part. But otherwise, let's get started. We have been looking at DNA replication. I have a quick overview. I think that you know most of this. I really advise that you go back and grab your Bio 1, whether it was from high school or from college, your Bio 1 notes when you started doing replication. For the first part of this, I think only one word is different. And maybe not, depending on who you had. We have 7 key steps in DNA replication we're going to need to talk about. We are going to talk about unwinding the helix, opening up the process so that we can access it with other enzymes. We're going to talk about reducing stress on that unwinding process so we don't break the DNA when we're trying to access the two sides so that we can create those complementary matches. We're going to talk about synthesizing a primer because DNA polymerases need a free 3 side to attach to. So I'm going to have to lay down something to prime an attachment spot. These 3 parts, 1, 2, 3, are initiation. We're going to initiate the process. And initiation really goes until we place the very first DNA nucleotide in our growing daughter strand. Then we're going to elongate, that's just 4 here, we're going to elongate the daughter strands, adding on to the 3' side in both of the parent strands as we complete our molecule. And then we are going to terminate the process. Termination, here, is kind of the wrap up and clean up. We're going to get rid of the RNA primers that we laid down. DNA polymerase 1 is going to replace them with DNA nucleotides. So the final molecule is not made out of RNA and DNA, just DNA. We're going to proof it, we're going to double check it, and then we're going to go back to interphase. The initiation of replication is encoded for by a protein, I think this is new to you, DNA-A. DNA-A is an initiator protein that is coded for by the gene DNA-A. This protein binds to the origin, and when it binds to the origin, it actually causes what's called a conformational change. That means a shape change in the helix, and that exposes little tiny strands of DNA. This is short strands. If you're reading this in your textbook, it assumes at this point that you know what all of these words mean. We talked about short strands of DNA when we talked about the DNA in general in bacteria and in other organisms. It's been a little while, so this stands for short strands. When DNA initiates the process by binding to the origin, DNA helicase tells you it's an enzyme. Heli tells you it's about the wow, that's not how you spell that at all. Helix. And DNA tells you the kind of molecule that helix is being enzymed upon. DNA helicase is made up of DNA B polypeptides, so these are proteins that are coded for by the DNA B gene. And this actually then calls that hollow enzyme, that huge machine that does the actual replication at the replication fork. I think this is new. I think that you have learned about DNA helicase in Bio 1, but I think you didn't learn about what tells it to come. As DNA helicase and that hollow enzyme assemble around the little short strand of DNA that's revealed, the process can be initiated. DNA-A, I'm leaving it up here so as a summary so you know where we've been. DNA-A binds to the DNA, revealing a little bit of a short strand of DNA to start the process. This recruits DNA helicase, which binds to that part of the helix. It's made out of DNA B polypeptides, so it's made out of proteins that are working together to function as the helicase enzyme. And that DNA helicase then calls this whole thing here, this hollow enzyme, which is going to actually cause the process of replication to proceed. As this helicase opens up the DNA helicase, so this part of the molecule is closed. This part right here is where DNA helicase is opening up the molecule. What would happen next is, because the two complementary strands that exist of DNA are held together by hydrogen bonds, and hydrogen bonds are sticky, what would happen is that molecule would immediately start wrapping itself back up again. We have to hold it open so that the process of replication can actually occur. That's the job of these single-stranded binding proteins, or SSBPs. The single-stranded binding proteins keep the helix open so the holoenzyme here that is the polymerases and all of the other processes that are going to need to happen have space to work. On the other side here, looking like a bead on a string, is a topoisomerase. This is DNA gyrase. I do not know if Bio 1, if you learned topoisomerase or gyrase. I will give you both. Gyrase is the specific one that we're looking at in bacterial replication. Gyrase is like gyrate here. Topoisomerase actually means the topology. Iso is same. It keeps the topology or the structure of the DNA the same, despite the fact that helicase is actually spinning really, really fast, like jet engine fast, as it unwinds the DNA. What would happen if gyrase wasn't there is, as helicase spun really, really fast, it would over twist the DNA in the opposite direction. And if you've ever been on a swing when you were a kid and you twist the swing until you're up on your tippy toes and you're spinning, and then you let it go, it'll unspin, but sometimes it'll drop real fast and you get that weird bottoming out feeling that's really what you're looking for when you're doing the spin. When that happens in the DNA tears. Gyrase prevents some of that stress, and if it does tear, gyrase can repair the tear, so it can relieve unwinding stress, but also it can reform the phosphodiester bonds. Phosphodiester bonds. If those break, it can relieve the twisting by relaxing it, but repairing the damage as it happens. After we unwind the whole thing, and it's twisting, we only want to unwind a little piece at a time that we can actually work on. RNA polymerase, so polymer means it's building a long chain. Ase tells you it's an enzyme. It's building an enzyme that builds a long chain of RNA. Primase is an RNA polymerase. It's called by helicase as helicase starts the unwinding function, and its function is to synthesize, to build a short segment of complementary RNA to the newly revealed open parent strands of DNA. This has to happen because the DNA polymerases that we have are unable to build without a free OH strand on the molecule. RNA doesn't have that rule. It can build by just making complements. So RNA builds a series of 20 to 50 to 150 nucleotide matches DNA to RNA. This is then later removed at the end of the process by DNA polymerase 1. This primer, we're looking at the DNA template here. We're reading the DNA template from 3 to 5, but we're building the new daughter strand, so this is new, and it's matching here. We're starting out by building an RNA primer with the help of primase. That leaves us a free OH at the end of this primer, where DNA polymerase can now build its new DNA matches to the template in the 5 to 3' direction. So I want to do a little bit of a review of how I would have taught this in Bio 1, because I think it's been a little while for most of you. This way, so if you are like, wow, I remember all of this, you can skip on, and I'll pause it. I'll leave a mark so that you can skip on to the next part. I want a quick review what I think you already know at this point, tying in a little bit about the new stuff. So the origin of replication is where we start the process. The DNA strands are separated and form a replication bubble. The replication process proceeds along replication forks. If you are drawing this on your own, two things that I am always suggesting for you: draw the replication bubble, mark the origin here where we are actually opening it up, mark your replication forks. So this is like a fork in the road, but you're facing the other way. This is the DNA replication is going to proceed this way and this way. This way and this way. And then I'm going to have you draw a line straight down the middle. Better than what I drew. So let me show you that. You have your old strands of DNA. These are your parent strands. It is G1, the very end of G1. And your cell just got a request from the body and is like, hey, cell, are you ready to replicate? And your cell assesses itself and it says, you know what? Yeah, I'm ready to replicate. Let's enter into S phase. And so the first thing you want to do when you start talking about it is figure out the directionality of your molecule. I'm going to make this my 5' side of this parent, which means that the sugars in this side are facing their 5' here. Out here is going to be their nucleotide, their nitrogenous base, and over here is going to be a phosphate group. Let's try to draw a phosphate group. This is the picture I gave you when we looked at this in the structure of DNA. We're looking at the 5, this is supposed to be a P, the 5' side here of the molecule. If this is 5, it means this side must be 3 because they all run in the same direction on one strand of DNA. Here's my phosphate. Here's my molecule. DNA is anti-parallel but complementary. So the anti-parallel means on the other side, all of the sugars are facing the other way. This is an adenine, but this molecule is facing its 3' side to the right and its 5' side to the left. CG, and then a little phosphate group up here. Establish directionality first, because if you don't have a direction, it's hard to answer or even think about the next step of the process. Then open it up in the middle. The replication bubble here is the place where we have DNA A has bound to the origin, which is very likely a series of adenines and thymines that are very easy to separate. It opens up the replication bubble with the help of DNA helicase, and then replication proceeds along the fork in both directions. So this direction and this direction away from the origin of replication. Draw a line down the middle, because when we think about replication happening in both directions, we actually are going to do it exactly the same but mirrored in each way. And we will do this in lab before spring break. I'm going to try to get it set up so it's kind of a tedious thing, but we're going to draw it out and then see what happens as we hopefully look at our glowing bacteria. When you worry about this, start by just worrying about one side of the replication bubble, and then as you get comfy, you can add in what's happening on the other side. We're going to draw out what happens from the origin to this side of the replication bubble, proceeding replication down the fork. We can ignore the right side for now, and we'll come back to it in lab, not in lecture. So here is my replication bubble made really, really big. This is the other side we're ignoring, right? We're not worried about it. We're going to start, this is 5, which means that my molecules in my parent strand are facing in this direction with their sugars. Their 3 side is, this means there's a flat side to the molecule here, and the 5 side, meaning the pointy bit with its phosphate, sticking to this direction. This here is the 5 side of the parent green molecule. This one is the 3 side of this parent green molecule. They're running anti-parallel, opposite each other, but they are complementary, meaning if this is a G, this is a C. And they form hydrogen bonds between them. DNA is, as it's opened, is unwound by DNA helicase. It breaks the helix and flattens it out, and then breaks the hydrogen bonds between the nitrogenous bases. A topoisomerase, DNA gyrase in this case, relaxes the helix and then can relieve the tension of helicase spinning, but also repair any damages to phosphodiester bonds that occur. Single-stranded binding proteins keep the strands apart. So if I drew them on here, here's my single-stranded binding proteins. I don't know how many there are, so this is not showing you something different here. My single-stranded binding proteins keep it open so that the hydrogen bonds between the different nucleotides do not just spontaneously reform. Here's helicase. Helicase is heading in this direction with the arrow. It's going that way. And topoisomerase is stabilizing this further down to make sure we don't tear the molecule. DNA helicase is breaking these hydrogen bonds. Single-stranded binding proteins are keeping them separated from each other. An RNA primer is required to put down a free OH. It builds a short strand of RNA. So this is my picture. And I always imagine RNA to be brown. I know that doesn't make any sense, but I always imagine it to be a brown structure. So I always color it in brown. RNA primase starts placing, here this is the 3 side of this parent strand. It places it, but an RNA, anti-parallel, running the opposite direction, but complementary. So if this was an A, this would be a U. If this was a C, this would be a G, and so on. And then RNA primase can build its first little place where there are free 3 sides. Look at that, a free 3 side with an available hydroxyl group or an OH for binding. We're going to remove this segment later with DNA polymerase 1 when we do our cleanup. DNA polymerase, once it finds this free 3 side, the holoenzyme that incorporates DNA polymerase 3 and the other molecules that are involved in doing DNA polymerization, building the DNA polymer, start to form new DNA pieces. Here, this is now DNA. I'm going to put a D in it for DNA. I'm very not good at drawing these molecules. These new DNA pieces are added, building on the free 3 side and elongating this strand. The first one that is placed is part of initiation. After that, we enter into elongation of the molecule. The daughter strand is elongated, building on the 3' side by DNA polymerase 3. When it does that, it's building phosphodiester bonds and elongating this strand here. We're not elongating the parent. The parent is already as long as it needs to be. As we build these, I pre-drew them, obviously. As we build these molecules, this daughter strand elongates, and it will continue to elongate. Helicase will move this way. Topoisomerase, the gyrases, will move this way. This entire holoenzyme will move. I'm leaving it simple here. We'll attach the other side in a second. The 2 strands of DNA are anti-parallel, which means that one runs 5 to 3 and the other one runs in the opposite direction. The DNA polymerase can only synthesize by building on the free 3 side, and that forms a phosphodiester bond between the previous 3 and the new nucleotide's 5' side. We'll talk about, I know that this sounds like 3s and 5s and 5s and 3s. I have puzzles from like 1962. They're actually pretty useful. We're going to try using them. What we just did was the leading strand. The leading strand is synthesized continuously, meaning that it's easy to build on the free 3 side. The other side of the molecule, the one we haven't drawn on yet, is the lagging strand. The lagging strand is synthesized discontinuously. What that means is it's synthesized in fragments. DNA polymerase III adds nucleotides to both the leading and the lagging strand. It works in the 5 to 3 direction. It can start building on a strand that has a free 3 side, but it cannot just build with no template, obviously, with no primer. RNA polymerase places the primer. RNA primase is an RNA polymerase that places the primer. There's a lot of P's in these sentences. The leading strand is what we just built. It's synthesized from 5 to 3, not the green parent strand, but the daughter strand. It elongates continuously. The lagging strand is going to go down here, and I've pre-drawn it, so bear with me here because I didn't want you to sit through me drawing. But I think it's helpful for you as well. There's very, very, very beautifully drawn pictures in the textbook and online, but you need to touch and draw this in order for it to make sense for your brain. You draw it out. Make yourself a bubble, make yourself a leading strand, and then ask yourself how we're going to build the lagging strand, because I have a big problem here because I'm elongating this way, but I can't go over here. Somebody else is working over there. That's heading off in the other direction. I need to build this strand, but this strand is going to run 3 to 5. And I physically cannot do that with my enzymes. We used to think in the 1950s and early 60s, we used to think that the molecule itself unraveled and then it built one way on the leading strand and then waited until there was enough space and then could build this way on the other side. That's why it's called the lagging strand, because it was initially assumed that it would lag behind the polymerization, the building of this leading strand. We know that is not true now. We know that it is built in short, tidy, not continuous fragments. Those fragments were discovered by 2 professors in Japan, Dr. and Dr. Okazaki, and they named it after themselves. Was their last name. They were a married couple. We need to go this way, and we need to build essentially and elongate in both cases to the left of my screen, but I can't build there. The molecules don't allow it. So the way we build, and I've pre-drawn this, so bear with me, is we lay down a series of primers. Each one of these brown one is an RNA primer. An RNA primase comes in and lays down a series of fragmented primers. It does everywhere. When the primer is there, this one has a 5 to 3. This one is 3 to 5. This one is 3 to 5. Those primers are facing the way that they are anti-parallel to the original parent strand, which means each one of them has a free 3 right there. This one has come up against the origin, so we'll ignore it. This free 3 is where I need to build because there's nothing here. So DNA polymerase builds 1, 2, 3, 4, and then it bumps into the primer. This is actually done by a series of loop-de-loops or back stitches. But for this example, I'm going to just say that we kind of take this molecule back, and then here is the next free 3 available. This would be 5, I can't count, 6, 7, here, 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 until we run into the next primer. And then we'll wait until the next RNA primer is built, 5 to 3. and then 7, 8, 9, 10, 11, 12 DNA primers until we run. So I build 1 Okazaki, 2 Okazaki, 3 Okazaki fragments. My overall effect of elongation is going to the left, but I'm actually adding nucleotides going to the right in this picture. So I'm still elongating with heading down the fork. I'm just adding everything in a little bit of a loop-de-loop or backwards. I gotta build on the 3' side. At the end of this process, we need to terminate all of the DNA polymerases, the gyrases, so the topoisomerase, gyrases, the helicases, all of these are going to fall off. And then an enzyme called DNA ligase glues it together. DNA ligase repairs the phosphodiester bonds. RNA is removed by DNA polymerase 1, and then ligase slides down the molecule and seals off any of the bumps or nodules that are sticking out so that the molecule itself is smooth and stable. And this aids in its ability to retain information because it has a very continuous and reliable structure. It's strong. In bacteria, there's actually a ton of different enzymes that are involved in DNA replication. I've only introduced some of them. We've basically called them by their product, their protein, or their role. We've talked about polymerase 1, polymerase 2 really briefly, polymerase 3, RNA primase, molecules that are involved in initiation, helicase, gyrase, which is the topoisomerase, ligase. Here, these are the genes that actually code for them. I'm going to ask you about the product or the role in exams. Mutations can 100% impair this replication. These are all genes. These are genes that are involved in copying your genes if you're a bacteria. Some of these mutations are famously temperature dependent, so I can control this process and cause bacteria to mutate by changing the temperature. It doesn't kill them or cause their insides to pop out, but it does cause them to make more and more mistakes. And because mutations are the source of allelic diversity, I can cause my bacteria to change more rapidly by interfering with this particular process and making them make mistakes during DNA replication. Or I can kill them, stop them from reproducing. So on to the stuff that I think is a little bit new for you. And if you want to take a break, that's cool. I'm done with the stuff that I think you know from Bio 1. I'm going to keep going because it fits really nicely here. I think I have about 5 slides of newish stuff for you. Because what you learned in Bio 1 was bacterial reproduction, not eukaryotic reproduction. A lot of it is the same. We're still got a double-stranded DNA. We're going to unwind it at the origin. We're going to form the replication forks. We're going to bidirectionally, so in both directions, go off and replicate the leading and the lagging strands. We're going to need polymerases. We're going to need the 4 nucleotide building blocks, so adenine, cytosine, guanine, and thymine with 3 phosphates attached. We're going to need a template, and we're going to need some kind of RNA primer. All of this is the same as in bacteria. But of course, it's more complicated. DNA in eukaryotes is just more. It's generally found in multiple chromosomes. Those chromosomes are linear. And that DNA isn't just hanging out as a nucleoid. It's wound tightly around little structures called nucleosomes. That's the next lecture. And we actually have multiple origins of replications. It makes copying massive amounts of DNA faster, but it means I have to have lots of different DNA replication holo enzymes working at the same time. When we look at yeast reproduction and DNA replication, and yeast are eukaryotes, so they have multiple genes just like we do. They usually have about 250 to 400 origin of replications, and they start at these lots of A's and T's repeating segments. So when I look for an origin on a yeast, and in a lot of organisms frequently, I look for these autonomously replicating sequences. They're ARSs. It's a sequence of A's and T's that identify the origin, and it seems to be conserved across the yeasts. They all have the same kind of repeating A's and T's. The origin is recognizable to us in the code. It's an A and T sequence because that's easiest to actually break the hydrogen bonds, as opposed to a sequence of C's and G's, which have more hydrogen bonds between them. In eukaryotes, we don't just start we don't just start replication by binding one single protein to the origin. We have a whole complex of things that are going to happen. It's called the pre-replication complex, or the pre-RC. If I haven't said this already, I feel like I have in some places but not others. Don't just memorize the short version as you're studying. Study the whole name. So pre-replication complex, the pre-RC, as a unit. There are especially in genetics, so many different little short versions of things, but many of them are the same. You have to draw them from context, what we're talking about. Learn the whole name, and then when you really, really are comfortable with what you're talking about, you can use the short version. In eukaryotes, the origin controls the start place and the timing. This replication complex are a 6-protein complex that tags the origin, and that origin builds up and other proteins come and bind to it to form an entire complex that is saying, hey, we are ready to start replication here. When polymerases actually initiate the synthesis, the origin replication complex is used to start the process. As that process proceeds and completes, the replication complex is degraded. It falls apart, it's disrupted, and then as we enter into interphase that's not ready to replicate again for a really long time, this makes sure, ideally, that cell doesn't initiate replication immediately after completing replication, or we're not immediately starting mitosis after going through mitosis. There's a time delay. In eukaryotes, because we open up multiple origins of replication and we replicate away from each one of them, there are multiple replicons. Replicons was the unit that was replicated together. If you're not sure, in video 1, prokaryotes had a single replicon because they replicated everything all at once. In eukaryotes, we replicate multiple directions and multiple replicons at a time. And maybe thousands of different polymerases working at the same time in order to replicate your particular DNA chromosomes and your code. We actually seem to have codes for 14 different polymerases. 3 of them are majorly involved in our DNA replication process. So if you look here, I'm not going to talk about all of these, but if you look here, there's a lot of, this is alpha, delta, epsilon, gamma, beta, eta, zeta, kappa, and so on. These are all different ways. I think this is supposed to be translation. These are all different polymerases that do different aspects of replication, including, kind of back to last week, repairing our mitochondrial DNA. Polymerase alpha has 4 subunits and low processivity. That means it falls off of the thing that it attaches to really quickly. This is a feature, not a bug. We're going to look at other enzymes that work this way. They grab a hold of the place where they're supposed to be working, but they're not great at staying on for the entire process. We want that with polymerase A. We don't want it to stay attached to the DNA and keep copying even when we're done with the process. DNA polymerase alpha grabs a hold and starts the process. It grabs onto an RNA primer on the leading and the lagging simultaneously and adds about 20 DNA nucleotides. Then it falls off. When it falls off, it's replaced by either delta or epsilon polymerase. Those have higher processivities and are more likely to stay on the molecule and complete their process ability to do their work. Polymerase delta and epsilon are higher processivity. They are really, really good at not just adding the DNA, matching the template, but they can also proofread and make sure that they're not making a mistake. We say that's exonuclease activity, or the ability to excise a nucleotide that is incorrect and add one that is correct. Polymerase delta here synthesizes the lagging strand. Polymerase epsilon synthesizes the leading strand, according to your book. I will say, and I made sure this wasn't on your homework, I think. I don't always see the exact questions you get, but I'm pretty sure we didn't talk about this in homework. This does not appear to be quite true anymore. If we look at the more recent data, it looks like delta actually plays a role in both the leading and the lagging strand. These are just newly understood. We are still learning a lot about them. So I would, in the back of your head, this is okay for now, but if this is where you're going and you're going to do genetics research, we're getting into the parts of genetics now where we actually aren't really sure what we're talking about. Not because it's college genetics, but because it's actually new information. This for sure is not quite right. It looks like if polymerase delta plays a role in both strands, polymerase epsilon may actually be doing more repair. Eukaryotic DNA is complex. It's built together with a lot of binding proteins. Way back, the first day of class, I think, we talked about the difference between chromosomes and chromatin. Chromatin is when eukaryotic DNA is kind of loose, it's hanging out, it's wrapped around proteins, but it's not wound real tight into chromosomes. When I look at eukaryotic nuclear material here, I see that this is the DNA, and then it's wrapped around these little tiny nuclear bodies, nucleosomes. Some is body. This is inside the nucleus bodies. This is where the DNA itself is wound around a series of proteins. This keeps it organized. It's very long and complicated. It also helps to keep it from tearing. But if I'm going to replicate this, I got to move those nucleosomes out of the way and just get at the DNA. And frankly, I got to copy the whole thing. I can't just skip over the bits where it's hard to copy because that information is essential, just not being used right now in this cell because it's wrapped tightly around a nucleosome. At the end of the chromosomes, at the end of each piece of DNA strand, there are telomeres. Telomeres are ends. They are generally not coding for anything, but they're kind of more like caps. They're made of nucleotides, but we think they're there to protect the ends of the chromosomes. And we've seen them when we looked at the Y and X chromosome, when we talked about sex chromosomes a couple weeks ago. They are repeating segments. We do not think they code for a gene. We think they are there to protect the ends. Because every time you replicate and you get to the end of the chromosome, you kind of fall off a little bit early with that holoenzyme replication machine. That means that the little bitty tip of each chromosome doesn't get copied each time. As that little bitty tip doesn't get copied, the chromosome over the generations of cells going through mitosis is going to follow a different thing, mitosis get clipped and shorter. The telomere has some T-loops and a complex of proteins that binds and shelters it, is a sheltering complex. Caps on the end. The enzyme telomerase repairs the telomeres as they get shorter. For most of your body cells, that is something that does not exist. So somatic means body. You don't have activated telomerase. We have studied in not me personally, but researchers have studied telomerase in cells of eukaryotes, and that repair in embryonic development makes sure that the tips of the chromosomes don't degrade. We see active telomerase in things like sea turtles and tortoises as well, it confers a sort of functional immortality to those cells. They can reproduce many, many, many more times without degrading their chromosomes than our cells can. If I add telomerase to my cells in an attempt to make them functionally immortal, you get cancer in mammals. It causes you to have cancer because those cells then can replicate without control. They'll replicate more frequently and they'll grow in a way that is not great for the living organism. I want to stress here, there's a lot of research and you'll see sometimes things being sold, like youth serums and things with telomerase in them. Probably not real, because real telomerase, if it was actually taken in, would cause bigger problems than it would help. It won't keep you young forever. It just gives you cancer. RNA is actually serving as a template for the synthesis of the DNA complement. This means as the primer. This in us, in eukaryotes, is a ribonucleic protein. It is made of RNA and protein. It's one of the few enzymes we'll come across that is actually an RNA protein enzyme. The other one is the ribosome itself. The RNA component is called TERK, or telomerase RNA component. It includes some reverse transcriptase. Reverse transcriptase means it can do transcription backwards. It can take an RNA fragment and turn it into a piece of DNA. We will continue in the next lecture a little bit about ribozymes, and then even further after spring break, I feel like a little bit more ribozymes. For this, we'll just sum it up with, I would like you to be comfortable with a little bit of the differences between eukaryotic and prokaryotic replication, polymerase alpha versus polymerase 1, for example. But overall, I'm holding you still to the process that bacteria do. Honestly, it's because the one in eukaryotes is less well understood, and I don't really have all of the solid answers in how eukaryotic replication works yet for you. I probably won't. You'll when you're teaching genetics, you can expand on that, I guess. So practice with me. What does RNA polymerase do in DNA replication? Anything? Yes. RNA polymerase is going to generate the primers in situ, in the cell, during DNA replication. We are going to change that when we get to PCR because we're going to do it in a machine. But if it's in a living body, the primers are made by RNA polymerase, a primase. DNA A, protein in bacterial DNA replication, is there to binds to the origin so that helicase can attach and unwind the double helix. It recruits helicase. Oh. Here. So, summary, I do not think that this is anything much new, a few new things. If we need to go over it a little bit more, the biggest part that catch people over the practicing from year to year is that 3 to 5 thing. I will help you with that and we'll build it. And then once we get that 3 to 5 thing, we'll be ready to start PCR and talking about the process of doing it in the lab once we understand how we do it in the body. And I'll see you guys in lab. I'll see you all in the next video, chapter 12. It's very short. There's only one video for chapter 12. All right. And hopefully it will be warm soon. All right. And from me and Alice, bye-bye.